Hi students, in the last class we discussed about different types of transgenic plants. Means we discussed about one of the pest resistant plants and insect resistant plants, right? And along with that we discussed the advantages of transgenic plants also. The advantages of transgenic plants also. And coming to the next topic is the applications of biotechnology in medicine field. Applications of biotechnology in medicine field. See, totally within medicine field, 30 therapeutic substances are synthesized by the biotechnology. In the entire medicine field throughout the world, 30 types of therapeutic substances are produced by the biotechnology. In that, 12 types of products are available within the market. Within the 30 types of therapeutic substance, right? 12 are available within the market. 12 are available within the market. Right? And the very well-known concept is the insulin production. The very first one is the insulin production. Not only insulin production, there is an interferon productions are there, and different types of hormones production is there. Right? So several types of products which are related to the medicine, which are produced by the biotechnology. See, here insulin production, before going to the production process, we already discussed. I given as examples so many times. Remember, in the olden days, the biotechnology is not available. Then we know some of the diabetic patients are there. Whatever that insulin dependent diabetes is there, means diabetes mellitus. Remember, one of the person having the diabetes disease is nothing but sugar levels raised within the blood. All of you remember, within our body at regular intervals, insulin hormone production takes place. And regulates the sugar levels. Once the insulin hormone is produced, the plasma membrane contains some of the glucose transporters, which are opened. Then the glucose molecules are entering inside the cell. Right? So generally, at regular intervals, the insulin hormone production takes place. Then the sugar levels are regulated within healthy person. And see, coming to the diabetic patient is there. Within diabetic patient, there is no production of insulin hormone at regular intervals. The sugar levels are raised within the blood. Then it's followed by causes several types of disorders. Right? See, here that is about the See here the history is initial days, the insulin production, the insulin extraction. Whatever the pig pancreas are collected, which are crushed, and insulin hormone is collected, isolated, purified, and that one is available within the market. See, that is the lengthy process and costly process. At the same time, within that process, whatever the waste is there, see, for insulin production, the whole pig is killed and the small pancreas is collected. So, if we follow that process, the waste production is more. And later, that process is replaced by the biotechnology. Within biotechnology, simply the genes are isolated, which are inserted into the bacteria, E. coli bacteria. Then insulin hormone production takes place, and purification process and the packing available within the market. So this is a very easy. This is not expensive. See here, insulin production, 1983. The scientist name is the done by the illegally, and he started one of the American company. So here we used modified E. coli bacteria. Okay. Early days, yeah, I discussed already. Early days, the insulin isolation or insulin extraction from the pig pancreas. Okay. See, we have to know the difference between inactive state of insulin and active state of insulin. That is nothing but pro-insulin hormone and insulin hormone. See, this is one of the pro-insulin hormone. Here this one having three chains. A chain is there, B chain is there, and C chain is there. See, the C chain is also called C peptide. So A chain, B chain, and the B chain. A chain having 21 amino acids and B chain having 30 amino acids. Right? And here there is a disulfide bonds are there between A chain and B chain. Two disulfide bonds are there. Within A chain, one disulfide bond is there. There is nothing but these are interdisulfide bonds, and this one is intradisulfide bond. These two are interdisulfide bonds, and this one is the intradisulfide bond. Totally three disulfide bonds are there. Right? During activation process, the insulin molecules are entered into the Golgi complex. See, we know this is one of the amino acid containing polypeptide chains, and these polypeptide chains connected, which are act like a hormone. So insulin is a polypeptide hormone. Polypeptide chains contain hormone or polypeptide hormone. So within the insulin, A chain, B chain, and the C chain. See, all these are formed by the amino acids. We know the activation of proteins. Takes place within the Golgi complex. So here activation of pro-insulin into insulin. Okay, this step is also takes place within the Golgi complex. Post-translational modifications are takes place within the Golgi complex. So here the cutting process within the human body, whatever that Golgi complex is there, the Golgi complex involved with the cutting of C peptide, right? Then this is the insulin. This is the active hormone. Then this hormone released into the bloodstream, binds to the insulin receptors, open the glucose transporters. Then the glucose molecules are transported from blood to inside the cells. Right? That is about the insulin hormone and the production. Right? See, this is not the production, the activation process. So how the insulin production takes place. Remember, in biotechnology chapter what we discussed, simply the insulin genes are isolated, the genes are inserted within the bacteria. Right? Then insulin production takes place. But that is not the exact right process. The exact right process is here. There is an A chain. The chain is responsible for the A chain gene. This is A chain gene. Right? The A chain gene is responsible for the A chain production. And this is a B chain gene. Right? So here there is an A chain gene, B chain gene. These are separately introduced into the vector. Again, the vector introduced into the bacteria. The bacteria is the E. coli bacteria. So here there is an A chain production, and here there is a B chain production which takes place. Both these are subjected for the purification process separately. After the purification, these are pull up, and these are mixed. Between these two chains, naturally there is an association is there. The association is nothing but disulfide bonds formation. Naturally these disulfide bonds are formed, then this is nothing but insulin hormone. So what is the difference between RNA technology produced insulin and natural insulin? Remember, here there is no pro-insulin. We are not producing here pro-insulin. In this process, directly we are producing insulin. Directly we are producing insulin. But in cells, there is a proinsulin is produced. The proinsulin is converted into insulin. The proinsulin is converted into insulin. Right? So that is about that biotechnology. One of the first application, production of insulin. Remember, in 1983, Eli Lilly introduced the biotechnology of insulin production, or genetically engineered insulin production, or recombinant insulin production. Right? That is about that the very first one, the insulin production. And next one we will discuss about the gene therapy. Next one we will discuss about the gene therapy. Right? And coming to the next application within biotechnology and related to the medicine field, gene therapy process. So what is gene therapy? What is gene therapy? Before going to the gene therapy, I like to discuss about chemotherapy. We know nowadays we are following the chemotherapy because any person had any disease. Okay, in that case, we are using some of the chemical components or tablets. These are nothing but tablets. We are taking some of the tablets. Presence of the tablets, most of the diseases will be cured. Most of the diseases will be cured. So the method is called as chemotherapy. The method is called as chemotherapy. And there is different types of therapies are there. Natural therapy is there. Natural therapy, right? And we know about homeotherapy, Ayurvedic therapy. So different types of therapies are there. Therapy is nothing but cures the disease. Cures the disease. That is nothing but treatment. Therapy is nothing but treatment. So gene therapy. Gene therapy is nothing but some of the genetical disorders are present within humans. Some of the genetic disorders are present. Some of the genetic disorders are there, right? And whatever the cells contains the genetic disorders are there. So there is an impairment in the genes. Impairment in the genes within the cells or within the person contains the cells. The cells, okay, there is a non-functional genes are there. Impairment is nothing but there is a non-functional genes are present. Within the person, there is a non-functional genes are present. Then, by the gene therapy process, we are replacing, replace the non-functional genes. We are replace the non-functional genes with functional genes. We have replaced the non-functional genes with functional genes. So, when the genes are defective, the enzymes are not produced, then the functions are not maintained. The state is nothing but disease. So here, we are replacing the defective genes. We are replacing the defective genes by the effective genes. That is nothing but non-functional genes replaced by the functional genes. So the concept is here, whatever that non-functional genes are present, which are responsible to cause the different types of disorders, right? We know about there is color blindness is the best example. That is one of the genetical disorder. And the finest example is the adenosine deaminase enzyme. Adenosine deaminase enzyme. See, within infants, one of the adenosine deaminase enzyme producing gene is there. Okay, some infants, this gene is not produced. Okay, this gene not functional. This gene is not functional. So, if the gene is not functional, there is no production of adenosine deaminase enzyme. There is no production of adenosine deaminase enzyme. Lymphocytes are not produced properly. Lymphocytes are not produced no lymphocytes production. Immune system is not properly maintained. Immune system is not properly maintained. Finally, that one is called as SIDS disease. The disease is called as SIDS disease. SIDS disease. SIDS is nothing but severe combined immunodeficiency syndrome. Severe combined immunodeficiency syndrome. Very simple concept. One of the young girl or young boy, within the young girl or young boy, there is a gene defect is there. The gene defect related to the adenosine deaminase enzyme. Right? If the enzyme is not produced, there is no proper production of lymphocytes. There is no proper production of lymphocytes. There is no proper production of lymphocytes. Right? Then immune system is not maintained. Immune system is not maintained. So that disease is called as SIDS disease. That disease is called as SIDS disease. So here this one is called as severe combined immunodeficiency syndrome. Finally, the person will be dead by the lack of proper immune system. Right? So by the usage of gene therapy in the 1940, a 4-year-old girl treated with ADA enzyme producing gene. ADA enzyme producing gene. Very simple. This is the one of the cell, and this is the nucleus, and this is the DNA, and this is the ADA producing gene. This is the defective gene. This is the defective gene. That defective gene is replaced by effective gene. The defective gene is replaced by the effective gene. The defective gene is replaced by effective gene. So the concept is very easy. Whatever the defective genes are there, the defective genes are replaced by the effective genes. So treated with ADA enzyme replacement. ADA enzyme gene replacement therapy. ADA enzyme gene replacement therapy. Right? So if the gene is replaced, then enzymes are produced, then lymphocytes are produced. There is no SIDS. There is no SIDS disease because within the person immune system is maintained. Within the person immune system is maintained. Right? And I like to discuss about the types of gene therapies. Types of gene therapies. Here, with gene therapy, two types of gene therapies are there. One is embryonic gene therapy. Embryonic gene therapy. See, we know some of the parents have the color blindness and some of the disorders. Then, initially, we did the genome analysis, the parent genome analysis. We find out some of the disorders. Maybe there is a chance. The diseases are expressed in the next generation. So in that case, what we are doing, see whatever that embryo is there, whatever that embryo is produced, the embryo is collected. Embryo is collected to that embryo, to that embryonic cells. We are inserting the effective genes. That is nothing but functional genes. We are inserting the functional gene. Once we are inserted the functional gene to the embryo, once we are inserted the functional gene to the embryo, so the treatment is the permanent gene therapy. The treatment is permanent gene therapy. The treatment is permanent gene therapy. Right, so concept is very easy here. Parents are there. Parents are having some of the genetic disorders. The disorders are identified. Right? Then there is a chance for the express some of the genetic disorders in the next generation. So in that case, okay, whatever the embryo is there, the embryo is collected within the laboratory. We are inserting the functional genes. Once we are insert the functional genes to the embryo throughout the lifespan, the functional gene is present in all the cells. So embryonic gene therapy is the permanent gene therapy. Right? So this is about the one type of gene therapy, that is embryonic gene therapy, right? Permanent gene therapy. And other one is the adult gene therapy. Adult gene therapy is nothing but, just now we discussed, there is an ADA gene, right? ADA gene, right? So suppose 4 years old girl, okay, as per the NCERT, 4 years old girl. So here there is no chance for the embryonic gene therapy. Then what we are doing? What are that person? Okay, lymphocyte producing cells are like lymphocyte producing cells are there. The cells are collected. And the cells treated with the ADA gene. Cells are treated with the ADA gene. Right? After that, again the cells are fused with, that is nothing but blood transfusion. Again the cells are fused. The cells are infused with the person. So within adult case, we have example, we are taking ADA gene and 4 years old girl, lymphocytes producing cells collected. In that cells within the laboratory, we are inserting the ADA gene, right? And after that insertion process, okay, again the cells are transfused into the person body. Then this process is not the permanent therapy. Because at regular intervals, okay, this process required. The patient required, okay, this treatment at regular intervals. So this is not the permanent therapy. This is a temporary process. Every 3 months or every 4 months, every 5 months or every 6 months, this treatment is required to the patient. Because lymphocytes have the lifespan. Once the lifespan is completed, okay, the cells will be dead. New cells are produced. Again, the new cells are not having that gene. Okay, the disease symptoms are appear. So that's the reason why. Okay, adult gene therapy process is not the permanent therapy. Okay, embryonic gene therapy process is the permanent therapy. This is the permanent gene therapy process or permanent treatment. This is the temporary treatment. Right? That is about the different types of gene therapies. Next one we discuss about the molecular diagnosis process.